this reference goes back to 1908, and I think somewhere around that time, the work of Mike Stiles started. Mike Stiles worked at the University of Edmonton, Alberta in Canada, and they designed the E. coli probiotic with the colicine produced, and that was one of the first tested in pigs, and they were so successful they started their own small company, and they were making these bacteriosis producing Escherichia coli for pig farmers, and I don't know if they're still around, they're still making it, but that was a very good example. Okay, fecal shedding, meaning that the microorganism persists in the environment of the eukaryotic host. Reduction of fecal shedding and colonization of O157H7, which is a really good example. And post-infectious period, the authors observed continuous shedding with the microorganism that was carrying the genes for the bacteriocin production. Two separate cattle trials, for example, and changing gears from colicines, which are produced naturally, to colicine next, which was engineered into Nielsen 1917, the microorganism you know. We were talking about it before, and I mentioned briefly when we were dealing with the engineering of bacteriocins that there was a specific standalone approach used to engineer Nielsen 1917 to become a more advanced probiotic microorganism. We didn't have time to talk about it. All right, two trials, significant number of newborn cows with diarrhea, and the diarrhea was used significantly with the colicin producing Nielsen 1917 from 65% to 26-12% respectively. I think this is a really decent biologically significant result. Another engineering bacteriocin produced lactose fermented colicin. Instead of diarrhea in pigs was reduced significantly compared to the control. Engineered E. coli cells producing micros reduce the numbers of salmonella in infected chicken to the undetectable levels. And you may remember we were talking about it that salmonella can be carried asymptomatically, but in some cases they can cause salmonella infections, can cause significant damage to the health of poultry. Control of mastitis, and mastitis is a huge problem, and the economical damage up to $1.7 billion to the dairy producers annually, just in the US. So this is a very big problem. And cows with mastitis randomly assigned niacin gentamicin treatment groups, and the intramammary infusion with either niacin or with gentamicin. And the clinical cure rates with niacin was about the same, 90%, over 90%, as with gentamicin. Niacin therapy eliminated 54% of staph aureus, and gentamicin eliminated 33%. And interestingly, that 80% of the isolates were resistant to penicillin, 35% resistant to gentamicin, but none of them were resistant to niacin. And I think this is a fantastic area where niacin can be used. And actually, I recall at least 5 companies internationally that are having natural products for treatment and prophylaxis of mastitis, where niacin is used in formulation as a natural additive. All right, so this is a very promising approach, but I have some ideas of how it can be used and how it can be improved. If Dr. Tiwari with us, we can discuss it in the future. Poultry, pediasin, niacin, some unidentified antibiotics and bacteriocins were used, bifida, lactococci, pediacocci, improvement of performance in the field trials, improvement of control of perfringens, and also elimination of the spores. In vitro, some data were in vivo later, but not reflected in this particular table. So good promises for bacteriocins in poultry as well, especially with perfringens, improvement of the performance. There were some isolated reports with no follow-up studies saying that niacin actually can also improve the weight gain. And I was very curious about these studies, weight gain in mice and in poultry, but there were no follow-up studies for both reports. Perfringens causes necrotic enteritis in chicken, we know that. And it can also be in the healthier birds, so the bird can carry it asymptomatically, all right? But if it causes necrotic enteritis, the mortality can reach up to 50%. And what is important here is prophylaxis, because once the infection develops into disease, there is no time for it, right? Causative factors are numerous, and of course, the microorganisms can come virtually from anywhere in the environment, and even if it is the bird is not dying, there can be a low performance, weight cannot be gained properly, and the economic consequences mentioned are up to $2 billion per year, and that's the loss to the producers, cumulative loss, if you wish. It's the death of the birds, it's the cleaning that is required for the facility, it's the assistance from the professional veterinarians, etc., etc. So the virulence and pathogenicity factors of perfringens and how they can be prevented, and you can see here stopping actions, and of course, vaccination, yes, it's very important. Probiotics, most definitely, yes, through the immunomodulation, through the production of bacteriocins. Adhesion of the pathogen can be interfered with, again, with the assistance of probiotics and with the assistance of bacteriocins through the quorum sensing. Prevention of the toxin production, again, quorum sensing can limiting the number of the pathogenic microorganisms. Bacteriophages are mentioned, lytic enzymes are mentioned, prebiotics as the factors that can inhibit non-desired microorganisms and improve performance of probiotics are mentioned as well. All right, so some of the examples of commercially available feed additives for the prevention of these infection in the birds, for instance. All right, and some of them have over 200 bacterial species. Quite frankly, I cannot believe in it, but well, that's what's on the label. Some have lactobacillus, some have yeast, and competitive exclusion in many cases is blamed as the major factor. In some other cases, probiotics are claimed as the major deliverable for the action. And in some instances, we have the immunomodulation and antimicrobial activity mentioned. And that's what we're doing with the yeasts in the formulation for the protection against the burns. Examples of these traits which can inhibit perfringens, and again we have bacilli, enterococci here, and some of the strains are coming from the broilers, healthy chicken from humans. The activity not tested, but mode of action in many cases, bacteriocins, either specified or non-specified. When we're coming to specified, identified bacteriocins for poultry applications, pidiocin, PA1, niacin, ruminococci, some antibiotics that can come from bifidobacteria, and all of them can effectively inhibit fringes in vivo trials, and we can see here improvement in the field performance, inhibition in vitro, etc. Growth promoting and antimicrobial effects of niacin. In another experiment, we have 300 chicks. 720 chicks, ROS 308, randomly distributed in the tested groups. Niacin, the source of niacin was Lactococcus lactis, species lactis. This is the niacin producer, and the supernatant contains a significant concentration of niacin in it. Treatments, there was positive control with ionophore coccidiostat. Salinomycin was used in that case. Negative control, no any treatment. Niacin was delivered to the birds. And as a result, take-home message, niacin effectively suppressed the microbial pathogenic activity in the gastrointestinal tract, improved performance, and the action was similar to salinomycin. I wonder, that's my question in the parentheses, if niacin is already or will be banned in Europe and perhaps anywhere else from the use as a prophylactic agent. When we were talking about niacin, I mentioned that there were no, for instance, niacin-resistant forms isolated when we were dealing with the treatment of mastitis. Now, the important thing, mastitis is a multimicrobial infection. We're dealing with multimicrobial infection, but for instance, we're talking about bacterial vaginosis. But there were no niacin-resistant forms isolated. However, the microorganisms can become resistant to many stressors of all the antimicrobial substances that antimicrobial peptides to which there were no reports on resistance built. I think meganin is the only one, and even though I still remember one report where the investigators managed to raise resistance against meganin, and they did it deliberately. So, how to prevent the microorganisms from becoming resistant to the stressor? Two approaches that come to the mind almost immediately. One is if you have a stressor with multiple mechanisms of action, such as many sanitizing agents. All right, but then with sanitizing agents, microorganisms often trigger the multiple stress response factors that actually are making bacteria resistant to other different nature stresses. But that's one and very valid and very effective approach. But we cannot use these kind of agents for animals or humans. Another approach would be to use formulations containing multiple stressors. And these multiple stressors will work on different targets in the cell. So, like, just kind of a level of approximation. If 1 in a million cells will become spontaneously resistant to a single stressor, we will need 10 to the 12 cells to spontaneously become resistant to 2 stressors with different targets. And what if we will combine more than 2? So we have niacin, a cute little doggy, we have cold, we have salt, additional barriers, and these are the bad guys who are trying to escape the reaction of our bacteriocins, our stress factors. All right, multiple hurdle approach. Niacin was used. There are more than a few examples, and this table just gives you a few, many, where niacin was used in combinations with ridurine, lactoperoxidase, lactoferrin, sodium diacetate, with bacteriophages, with high hydrostatic pressure. And what you see here is significant reduction of listeria, 5-log reduction. Synergistic action against Staphylococcus aureus, extension of the shelf life. Now, beautiful example where niacin and lactoferrin can inhibit O157H7. And this is really fantastic because niacin on its own will not work against the gram-negative microorganisms. So you need something to disrupt the outer membrane. The inner membrane, gram-positive bacteriocins can't deal with, but you need to get into the inner membrane. All right. So niacin and lactoferrin work also synergistically on inhibition of Listeria monocytogenes, which is a gram-positive microorganism. So a variety of formulations, physical methods, other antimicrobial proteins, plant extracts. And these are just a few approaches where niacin can work synergistically, and we should distinguish synergistic action, additive effect, and in some cases antimicrobials can antagonize their action. So clever, intelligent approach in the design of multiple hurdle formulation is the must, right? So different targets and ability for them to coexist and to work together is important. Another example, and again it comes from somehow our work, Lillian Nielsen, the first author of this paper, almost a quarter of a century back. Wow, time is flying. She came to our lab from the Danish Institute for Fishery with the objective to find the ways to control pathogens and spoilage microorganisms, but mostly Listeria monocytogenes, in the smoked salmon. And they wanted to do it in the most gentle manner. So what we did, we studied niacin, carbon dioxide, and cold as the factors, as the stressors to effectively control Listeria monocytogenes. And because of the time constraint, I cannot show you all the experiments we started with the mechanistic studies. We showed that the membrane composition is changing because of the influence of low temperature and carbon dioxide. It is changing even the way that niacin-resistant membrane becomes sensitive again, and becomes sensitive because the cell must maintain its normal physiology under the changing conditions of the environment. So changes the membrane that becomes sensitive to niacin. So all three factors work really well as synergistic factors that we studied it mechanistically, and we studied it also in the formulation against cells, and obviously Nielsen studied it further in the actual products. And one of the examples would be the ACMA smoked salmon, which is a product of Denmark, and you see niacin is in the formulation. And I recall for some years USDA was using this paper, this study, as an example on their web page. But do we always need to kill the microorganisms to protect our environment? Can we protect the environment by controlling the biofilm formation? And I guess this is a very potent and really promising approach, which can be used and is used in a variety of scenarios. And this is where I want to change the gears again and give you my beloved Subtolocin A with no less beloved muscle subtilis Catmira 1933, the microorganism we were talking about before in our probiotic sessions. And subtilosin A is a very well-known triosine 35 amino acids, post-translational modified, cyclopeptide, group 1 triosines according to the classification where we have also in the subgroup niacin, temperature stable, active against a variety of pathogens. An important thing, it does not kill healthy lactobacilli. And that is why it works beautifully in the formulation for vaginal control of bacterial vaginosis associated microorganisms, living alone, healthy, live and happy vaginal lactobacilli. Now, when I started working with these, I read one of the papers from the gurus who published on subtilosin, and one of the papers was actually a death certificate. It was saying, well, okay, it works against such and such pathogen, so what's new? Who cares? There are so many bacteriocins that do this kind of job, and there is no future for this molecule. And I said to myself, well, we probably should start working with it for one at least reason, that it does not kill healthy lactobacilli. It's a selective action. Good. And so what we see is, of course, listeria is the gram-positive microorganism, and we see the concentration-dependent, reduction of viability of listeria by subtilosin. And so from these we can select the concentration which doesn't kill the cells at all, and they grow to the numbers sufficient for biofilm formation. But, and this is the control, of course, no subtilosin added, but with addition of subtilosin, still the concentrations that permit the same numbers of seemingly prosperous listeria have effective inhibition of the biofilm formation. And again, because of the time limitation, I have no ability to talk about the actual mechanistic study, but using the Vibrio cells, we showed that there is an inhibition of quorum sensing signals by subtilosin molecules. Doesn't inhibit the growth of E. coli at all, but we can see that it inhibits the biofilm formation by E. coli cells. Escaping the foods, where else? There are some places to go. And again, Gardnerella vaginalis. We were talking about it before, and we compared mechanistic studies where we looked at how it works against the cells. And we didn't see the dissipation of the membrane potential, and of course, valinamycin is a reference molecule which causes almost immediate dissipation. And niacin causes immediate dissipation of the membrane potential. This is where niacin added, and it works almost instantaneously. But not in the case of subtilosin, but it kills the cells. How does it kill the cells? And we looked at the effect of the delta pH. And certainly, immediate drop, subtilosin added an immediate drop in the delta pH. Nitricin had no effect, indicating the dissipation, of course. And niacin caused some gradual dissipation. Here is niacin added and gradual dissipation. And of course, nitricin didn't have any further effect because complete dissipation was achieved with niacin. So dissipation of delta pH with assistance of subtilosin can be achieved. But it's not only that. Subtilosin is acting against viruses. It's active against herpes simplex virus type 1. It's active against herpes simplex virus type 2. It is active against the viruses that are resistant to antiviral agents. Of course, the concentrations are somewhat high, but it's good that we have at least in our pocket something that may act when the antiviral agents are failing. The spermicidal activity was reported by others to subtulocin A, and it immobilizes human spermatozoa, and this is happening, you can see in the concentration-dependent manner. It also decreases the viability of the sperm, and again, you can see it's like control, and we're going to high concentration, and again, the decrease in sperm viability is a concentration-dependent process. And in the past, these experiments were done with the bovine sperm, but we worked together with some medical professionals, and we did these studies on the human sperm. And again, it's a very good result, indicating that perhaps it can be used as antibacterial, antiviral, and spermicidal agent. And so we're presently at the crossroad. Okay, so where to go and what to do next. And with this crossroad situation, we need to take we have to zoom out and to take the look at the bacteriocins, what they are, what they can do, how can we benefit from them. And again, you already know that I like these pseudo-linear type of graphs. Bacteria, viruses, eukaryotes, and concentration of bacteriocin is plotted here at the smaller concentration. So we have interspecies and intraspecies communication, which is reflected in quorum sensing and quorum quenching behavior, promotion of signaling and arresting these signaling processes, communication with the brother and sister cells, giving positive signals, and repelling, if you wish, the intruders who may be coming to the environment. With a growing concentration, bacteriocins can work as bacteriostatic, cidal, or lytic molecules. And I'm not talking about the primarily mode of action, because some of them are primarily bacteriostatic or cidal or lytic. With the same niacin, it can work at a low concentration by limiting the growth of the population, killing cells, or even as a detergent. They work against eukaryotic viruses. They can be spermicidal, and there are reports on bacteriocins specifically targeting cancer cells, eukaryotic cancer cells, and not killing the healthy eukaryotic cells. Of course, concentrations are rather high, but I don't know, maybe these are the kind of molecules that can be used either, in their natural shapes, or maybe these molecules can be used to work on lipidomatics and create new generation of artificial synthetic molecules, learning from bacteriocins action against specific action without side effects action on the cancer cells. Saying again a few words about the food, because food is something that is important for us here. And when we were publishing these, I was trying to be humorous. Of course, functional flavors, of course, low water activity. What kind of challenging microorganisms? How many of them you can expect in this kind of product? So I was trying to be humorous. Scientifically proven, peer-reviewed. Anyway, cheese and sausages are more likely to be the target for the delivery. They can be partially purified or fully purified bacteriocins. They can be delivered with the fermenting microorganisms or protective cultures, which are not involved in fermentation but sitting there and with some limited metabolism and activity, making bacteriocins and other substances to protect food environments. They can be delivered in the purified form, which is highly unlikely that they will do something. They can be encapsulated in a variety of ways. Some of the delivery vehicles are, depicted here. They can be delivered alone, or they can be delivered in a combination and a formulation with the synergistically acting substances, which for me is perhaps the best way to go. Don't forget to say a few words about controlled delivery. Yes, controlled delivery, bimodal delivery, which can be achieved, which is the most effective, not only in medicinal applications and pharmaceutical, which is known for ages, but also in the food systems, and that's what we showed, and this is how it must be done. Boost with the big concentrations first, and then smaller concentrations over an extended period of time. And this is the most effective delivery for antimicrobial proteins. Paper published in 2012 by the group which I really very much respect. Probiotic trait. I would cross it and I will add role in pro and eukaryotic ecology, because I think this is very important. This is what is missing from understanding of the future of bacteriocins. Probiotic functionality, how they act on colonization, how they act as killing agents and eliminating pathogens, how they act as signaling molecules, either positive signals or negative signals. And we're now at the crossroad. Not much is happening in a sense of applications, okay? And these studies started in 1925, and we have some, latest examples of the result. The USDA, approval breast status 1988, when 2023, where are these new formulations with bacteriocins for food applications and medicinal applications and over-the-counter products? I don't see them. What is happening? Where are they? And some thoughts from my personal humble experience, and of course I share it freely because we're all scientists, we all need to communicate and to work together. Fundamental research, translational studies and applications. There is still place for the discovery of novel bacterials. There is still need to study their structure-function relations. There is a desperate for the study of the role of bacteriocins in ecology. There is still a need in study and engineering bacteriocins for targeted delivery and targeted action. I have to mention here the fantastic work that's being conducted by many scientists, but most of all the groups from Tiagask and University College Cork in Ireland. I already mentioned peptidemimetics, and I did work on peptidemimetics for some time, and I think this has a future. We use the bacteriocins to design, based on their structure and function, to design new synthetic molecules to control vaginal pathogens, which was a very interesting work on its own. Translational studies, bacteriocins in multiple hurdles, there is a desperate need to continue. Again, natural peptidemimetics and targeted engineering, and again, ecology. I cannot emphasize enough ecological studies. There is a niche, it must be done. All right, and of course applications, food preservation, personal care products, medicinal applications, and again, ecology. Since a lot of my studies were also involved in these final gathering report, I have to mention some granting agencies, NIH, USDA, Bill and Melinda Gates Foundation, some collaborators and former students here are also mentioned. Santosh, you're here as well as my collaborator. Thank you for being with me. Thank you for being a part of the family. And it's a pleasure and honor knowing you and all of those I was working with through all these years. And thank you very much for your attention. And with these, I will finish our last session, and I will finish our gathering. I would like to thank all of you for your patience, for being with me, for being with us through all this time, and I particularly appreciate you being with us today, because today is holiday, national holiday, and thank you very much, all of you, and I'm open to the questions. Yes, there was a question about how we can get into collaboration, working together. This should be addressed to Dr. Tiwari.
Dr. Chikindas, I think in validity session we will address this at the same time.
Yeah.
So this question may be interacted, but you have taken very rigorous 2 hours classes, so we really apologize that continuously you have taken, and because our guests have joined and we have to start our Thanksgiving session. You know, if you start something, if you start something good, you have to end somewhere. And I am punished to organize this ending program, which is a painful for me. And sometimes you have to do it whenever you start. And we are waiting our Dean Academic Affairs. He has not joined. He has just given a message. And it was a very good information we had in last 5 days with Dr. Chikindas. Our chief guest, Professor J.P. Yadav, he has joined. He is a vice chancellor of a nearby university we have here, Indira Gandhi University, Mirpur. I request him to join the DIAS, virtual DIAS we are having rather. And we are having our Dean College Development Council as a guest of honor. We are having with him. I request you, sir, to join with us on this occasion of validity function. And we have a guest of honor, Dean Academic Affairs with us. And he is just connecting, probably will join very soon. And I invite local coordinator, Professor B. Narasimhan, also to join with us to start our valedictory function as a kind of Thanksgiving session for our foreign faculty member. So, Professor Narasimhan is our guest at mine.
I think so, sir. We have already joined. Just we can confirm Dean, sir. Others have joined.
Surender sir, Dean Academic Affairs, or probably he told me that just I am joining. So, any interaction or query from the participants? We apologize that we have not taken question this time right away, but you can, Professor Chikindas is with us, and you can interact in the last of this session. You can ask the question and interact also. We have interactions also.
Sure, and of course, all of you, it doesn't have to be today, but you can also communicate anytime with Dr. Tiwari, and we're staying in touch on a regular basis, still working together, and hopefully we'll be working together on numerous projects, and of course, we're open for communication and collaborations. So if you have any questions, if something will come to your mind later in time, just drop the line to Dr. Tiwari, and we will do our best to respond to your questions. So we're waiting now for the respective committee to join us?
Yes. Most of us, we have joined, but our Dean Academic Affairs is going to join very soon.
Fantastic. Yeah, and I've got a message. I had even a chance to communicate, and that's my usual life. 4:30 in the morning, my time, I was communicating with Dr. Todorov, who traveled now from Brazil to Amsterdam, where he is participating in the probiotics conference. that is organized by Dr. Weinemann. Yeah.
Okay.
Tiwari sir, sir has joined, I think so. That all is online only.
Okay. Okay. So, good evening to all of you, our respected dignity on the dais, and Professor Michael Chikindas. With all your due permission, I am starting this valedictory and Thanksgiving session to our foreign faculty. And I invite Professor B. Narasimhan for formal welcome to the gathering. Sir, please.
Thank you, Tiwari sir. Good evening, everyone. I take this opportunity to welcome our foreign faculty, Professor M.L. Chikindas, who has given us, we can say that, a lot of experience and knowledge sharing with our research scholars. So, for over a period of from 7 days, that I am happy to welcome you and thank you for giving us an opportunity to interact with you. So, I hope all the participants have benefited by with the interaction or the ideas you have shared, so they can utilize them for their future research and new research ideas might be generated among our research scholars and the faculty members. And definitely, we have the opportunity to collaborate with you through Dr. Tiwari, so person who scholars and research faculty, we will interact with you. I welcome you, sir, for this valedictory function, and I thank to that they thank our honorable Vice Chancellor Professor Rajveer Singh, who has uh, due to his some commitment is unable to join today the degree function, but I thank for him for his extended support, and I welcome Professor J.P. Yadav, Vice Chancellor, Indira Gandhi University, Mirpur, chief guest of the function, guest of honor Professor Surendra Kumar, Dean Academic Affairs, Professor A.M. Dean College Development Council, and all the HODs and directors are deans, those and the faculty members of MDU and from other participants who have joined this valedictory function, especially the participants who has gained a lot of knowledge from Professor Kindat and the local coordinator, the course coordinator Tiwari during the tutorial session. I welcome you all for this panel session, and I hope you might have enjoyed this 1 week session of your that GIAN program, and it is the very good opportunity provided by the Ministry of Education to interact with the foreign faculty and to gain the knowledge, because when we are going out of box thinking, we need some guidance from experienced person, and that is we can say that it is Professor Melvin as the right person is having what experience. And I welcome you all for this what is considered as inaugural valedictory function. I also take this opportunity to welcome our course coordinator, Dr. Tiwari also. So thank you. I hope you might have enjoyed. So now I hand over to Tiwari for further proceedings.
Thank you, sir. Thank you very much for nicely starting the function, and also welcoming all our guests who are present there. Before inviting our guests for their delivery, I would like to put some highlight on the course delivered by Professor Chikindas in last 5 days. So on first day, his lecture was on probiotics and general introduction in the first lecture, and the second was mostly on probiotics for gastrointestinal tract health. In the tutorial session, the students will get exposure for the instrumentation virtual with Department of Microbiology, and also the instrumentation technology in the MTech Chandigarh we connected on the virtually. Participants got exposure for that also. And the next lecture was probiotics for urogenital tract and oral health, and probiotics in pediatric health development and challenges. On that day, the tutorial session was a kind of virtual visit to a very related industry. We have Yakult at the Sonipat. So they demonstrated the students that how the product can be developed at the industry level, and the students got a kind of interaction with the industries also. And there was some virtual visit from the microbial genetics laboratory from Department of Genetics itself regarding several instruments which can be utilized for the work of probiotic experiments. Dr. Chikinda's next lecture was probiotics, microbiota and obesity, probiotic functional genomics and engineering. And the tutorial session was organized in the laboratory of Central University of Haryana, where they were connecting with the artificial gut system to work some experiments in the in vitro, rather in the animals. So they got with the interaction and also a laboratory demonstration, live laboratory demonstration in the microbial genetics laboratory in MD Guruvardhan. His next lecture was probiotics in agriculture, and also a very good design basic concepts on prebiotics, probiotics, postbiotics, and he clarified everything over us also to understand the concepts may be clear from all the students. And we had some purification and probiotic growth conditions optimizations demonstration from the laboratory, our laboratory also. His today lecture was in the probiotics formulations and dietary, and also on the bacteriosense. We connected today in the tutorial session with the Niti Aayog Government of India initiative at Atal Incubation Center, and the CEO of the Atal Incubation Center is directly connected with the participants and solved several issues how this probiotic product can be taken to a great height into a commercialized product. In this program, there are 68 participants have been registered from different states of India, including Haryana, Rajasthan, Karnataka, Delhi, and Maharashtra. And all the students being a virtual mode, they were highly benefited. So these were the short brief highlights of the program Professor Chikindas connected. So we were able to deliver the participants not only a theoretical lecture by a foreign experts, they were also able to interact with the foreign experts, and we had a visit to the industry, to the government initiative and the academia. Also, along with the instrumentation center we have, which can help such type of research in future. So with these words, I would like to move ahead and invite foreign faculty himself to have a concluding remarks over it, and a kind of possible collaborative opportunity in the future if we have, by Professor Michael Chikindas, Tadgar University. Over to you, sir.
Thank you, Dr. Tiwari. I would like to start by expressing my gratitude to thank the founding agency for putting a significant effort into making this event happen. I would like to extend my gratitude to the university administration, because this is very important for all of us to have a support, to have a feeling that what we're doing is understood by our senior peers, and they give their blessing to us. And this is an enormous encouragement. Winning the grant is one thing, but feeling that you're encouraged by your senior colleagues is enormously, and I would like to expand my thanks to the senior administration of the university. I would like to thank the technical staff. There were a few small glitches, I don't know, on my side, on the other side, but they were really very few, and I hope they didn't influence badly the course of the events. I would like to thank the technical staff for their enormous job on making it happen and making it run smoothly. And I know that we have like more than 60 people from all across country participating in this event. I mentioned during several sessions that while some of the information is a textbook material, some information comes from recently published papers in 2020s, 2023, but one of the objectives was not just to share the knowledge from the published papers and books, but also to ignite interest, to provoke participants to think outside the box, to see the things, the white spots, something that is still unanswered, something that still requires further investigation, or perhaps even to see the fields where they can apply their knowledge and all their talents to develop the science and to develop new applications. So if I manage to do that, that is the most satisfactory for me. We try to cover the various aspects from human applications to animal applications. We try to look at what these molecules are, what these bacteria are, and how they should be or can be delivered to the site of action. And all of these spheres, all of these modules of the course, I think each of them deserves to be considered as a separate opportunity for future collaborations and future developments mutually beneficial and fruitful relations. This effort didn't come from nowhere. We did have a very good foundation built with Dr. Tiwari over many years, if not decades, of fruitful collaboration, which started with Dr. Tiwari visiting our laboratory some years ago. And it wasn't just a single event. It turned to be into almost a family-like relation, and that's how I feel for us, being a family in research, in thoughts, in desires, everything we want to achieve. So with confidence, I can say that I believe that this gathering did serve multiple purpose, educational purpose, provocative purpose, to provoke the thinking process in the younger generation and other scientists, and as a catalyst for the continuation of our successful future collaborations. And Dr. Tiwari and I already exchanged several ideas. Some of them are probably crazy, some of them are not very crazy, but the ideas about the possible future directions of collaborations in health-promoting bacteria and antimicrobial proteins. So with that, I hope, at least I'm left with a feeling that this course really served several purposes, and they were really very good purposes for the entire group and for the future developments. And again, I would like to expand my thanks to all the participants, especially those who came today. I know we have around 40 people who joined us today, although there is a national holiday. Thank you very much for your participation, for your inspiring questions, and for being with us. And I hope that some of you will continue collaborating with us in the future. Thank you.
Thank you very much, Professor Chikindas, and your words are motivational for us, and the suggestions will include in our policy making, our ideas. And in this order, we have guest of honor with us, Dean College Development Council, Professor A.S. Mann. He is having with us, and I would like to invite his words from his side for the benefit of our audience. Professor A.S. Mann, sir, over to you.
Thanks, Dr. Tiwari. Am I audible?
Ji, sir, you are audible. Please continue.
Actually, I switched off my video for the sake of clarity. So,
no
first of all, a very good evening to one and all. It is indeed a matter of great pride and privilege to be part of this valedictory session of the 1-week GIAN course entitled Translational Science for Health Problem health promotion, food and feed, sponsored by Ministry of Education, Government of India, and organized by MDU Rohtak. I take this opportunity to heartily welcome Professor M.L. Chikandas, Rutgers University, USA, and at the same time express my heartfelt thanks and gratitude for him to be part of this initiative. I also wish to convey my greetings to Professor Surinder Kumar, Dean Academic Affairs, MDU Rohtak, and my dear friend Professor J.P. Yadav, Vice Chancellor, Indira Gandhi University, Mirpur, Rewari, who are on board in this session. The topic or the theme of this course is in great synergy with the thrust area of Maharishi Dayanand University, as Professor Chikandas has just revealed about the work done by Dr. Tiwari. In addition to which, various serious efforts are being made in various subdomains of life sciences, and it is also worth mentioning that a large number of my peers have been able to contribute significantly. Moreover, the relevance of the theme could not have been more than apt from the societal context, be it the countrywide or the worldwide, in view of the ever-increasing population or depleting natural resources, or as fulfillment of sustainable development goals. Such activities in longer term are surely going to be path-breaking as a result of the confluence of ideas and realization of realistic goals. I was just hearing Professor Chikandas about certain very important points he has raised regarding out-of-box ideas and realization on ground of these ideas. While sharing all this, I am indeed thankful and express my gratitude to the dynamic and visionary leadership of our Vice Chancellor, Professor Rajveer Singh, who is always there to inspire each one of us to be part of one or the other noble initiative. I would also like to congratulate Professor B. Narasimhan, Local Coordinator GIAN, and Dr. Santosh Tiwari, Course Coordinator, for all the hard work and meticulous planning in organizing this activity successfully. My last but not the least, my congratulations to all the participants for the successful completion of the program, and I feel encouraged by the concluding remarks of Professor Chikandas about the vast opportunities which are there in form of collaborative work, and I really look forward to the same from our university perspective. Thank you all. Good night.
Thank you very much, sir. Your kind words are always motivational for us and the university. We have a guest of honor with us, our Dean Academic Affairs with us, Honorable Professor Surinder Kumar, sir. And I invite him to say a few words on this dais. Welcome you, sir, on this platform of valedictory function.
Professor Tiwari, I am audible?
Yes, sir, you are audible. Please go ahead.
Namaste, everybody. I am Surinder Kumar, Dean Academic Affairs, MD University, Rohtak. In this valedictory session of this GIAN course, I welcome you all who have joined online platform, and especially I thank Professor Chikandas, under whose able guidance and leadership this one-week course was conducted. And today we are assembled on this online platform for the valedictory session. Thank you very much, Professor Chikandas. And I also thank Professor Tiwari, local coordinator, course coordinator, and Professor B. Narasimhan, who is local coordinator for this program. As we have Professor Maan with us, our Dean Colleges, and Professor J.P. Yadav, Vice Chancellor from Indira Gandhi University, Mirpur Rivari, and all other participants who have joined this valedictory session. I welcome all the participants. As Professor Tiwari has given some highlights of this about this course, so we know that under the guidance of Professor Chikandas, our participants have learned a lot. As I am not a student of science, I am a student of philosophy, but I understand that this translational science serves as a critical bridge between scientific discoveries and practical applications, playing a pivotal role in advancing health promotion and enhancing the quality of food and feed. As we all know that we are around 7 billion people on this earth, and health is primary concern for all of us, and for that health, food is a basic need, a healthy food. And without research and researchers, we cannot have healthy food for all the people. So this translational science, this multidisciplinary approach facilitates the seamless integration of knowledge from basic research into real-world solutions. And under Professor Chikanda's guidance, our students, our researchers, our faculty members from almost many states from the country, as I have the number, 68 participants have participated in this program from Haryana, Rajasthan, Delhi, Karnataka. So our researchers have a very good insight under the leadership of Professor Chikandas. So I thank you very much, Professor Chikandas. My heartfelt gratitude to you, and Professor Tiwari also, who is the course coordinator of this program, and Professor B. Narasimhan. And I also submit my gratitude to our Vice Chancellor for these kinds of initiatives under whose leadership we are progressing day by day. So in the end, I welcome and I put my thanks to all the participants who participated in this one-week program. So this is all from me. Namaste. Thank you.
Thank you, sir. Thank you very much. Your kind general suggestion has kept us a very high, despite of the boundary of science and art. It doesn't matter. It's a motivation and the inspiration we get from you. It always works us to do great thing in our life. Thank you very much, sir. With this order, I invite Chief Guest of the Day, Professor J.P. Yadav, who is Vice Chancellor of Indira Gandhi University, Mirpur. Professor J.P. Yadav was instrumental in the submission of our GIAN program also. At that time, he was working with us at our department, and he was kind enough to support most of the technical part to prepare this, to prepare the proposal and design it, submit to the funding agency, communicate. And in his guidance, actually, we planned this course during the COVID time, and due to some emergency at that time, we were not able to conduct this program, and everything was set up, but due to that emergency, we had to fail to do that. So this is the reason he is very linked with the genesis of this submission to the funding agency of this course, and he know the background better than me. And I would like to invite our chief guest, Professor J.P. Yadav, to deliver his views and blessing to us. Professor Yadav, over to you.
Thank you, Dr. Tiwari. Our respected, learned and world-known phone expert, Professor M.L. Chikandas from Rutgers University, USA. Dean Academic Affairs and guest of honor on the valedictory function of this GIAN course, Professor Surendra Kumar. Guest of honor, Professor A. Man is also having the charge of Dean Colleges. Our local coordinator, Professor Narsiman, and course coordinator, Professor Santosh Tiwari. Other faculty members from the different departments of MD University Rohtak, and the participants from Haryana, Rajasthan, Delhi, Karnataka and Maharashtra. A very good evening to all of you. This one-week GIAN course is conducted at MD University Rohtak on translational sciences for health promotion, food and feed. And this course is taught by Professor M.L. Chikandas, who is world-renowned expert in the field of probiotics and bacteriocins. You can judge his expertise because he has a citation of more than 14,000 and his H index is 56 and I-10 index is 144. So we can judge our expert is so much learned, and certainly he taught this course in different 10 lectures which cover the major aspects of modern development in the field of health, promoting natural derived formulations and their application in human health, personal care, functional food and feed. The probiotics, as I have just noted from the lecture of Professor M.L. Chikandas and summarized by Dr. Tiwari, these probiotics, they have a wide application in the human health. It covers in the different aspects as a antibacterial activity, application in pediatrics. Even beside the human health, the probiotics has also application in the agriculture field. So this course has provided a basic platform to train the participants in identification of the project related challenges and ability to solve research related problems. And the GIAN course was sanctioned when I was local coordinator at MD University Rohtak under the leadership of Professor Rajbir Singh, Honorable Vice Chancellor of MD University Rohtak. 6 of the courses they were already completed, and this was the last course that is completed now today. Earlier, before the COVID, Professor M.L. Chikandas supposed to come physically, but at that time COVID has come and we are unfortunate that Professor M.L. Chikandas he didn't come to us at this MD University Rohtak in physical. But somehow this course is completed online, and all the participants, they have got benefited from the expertise of Professor M.L. Chikandas. And the GIAN is also have the different aims that under this GIAN program, reputed international faculty that can interact with the teachers, with the students, so that our faculty members and students, they can get exposure and they can get experience from reputed international faculty, and check out the collaborative research program, and may also having interaction with the industry people, so that some entrepreneurship can be taken. And I hope some of the participants certainly will go for entrepreneurship, because the probiotics has a wide applications. And you may have, by establishing the entrepreneurship, you will be job giver instead of job seeker. And one more thing, that there are number of application of probiotics. Some researchers, and I also asked Professor M.L. Chikandas also explore the possibility of these probiotics for antiviral treatment, such as the COVID type viruses. So these applications can also be explored by using these probiotics. And I congratulate the participants, those who have completed this course, and I also congratulate Professor Santosh Tiwari that he has taken the task to complete this course through the online mode. So with these words, my gratitude to all of you, and I hope certainly you will have benefited by the guest lecture delivered by Professor M.L. Chikandas. And I also thankful to the university administration and Professor Tiwari for inviting me on this occasion. Thank you, all of you. This is all of from my side. Thank you, all of you.
Thank you very much, sir. Thanks for kind words. The idea you generated that time has come today a reality. And this is the only reason Professor Chikandas and us all are here on this platform and conducting this course, only because of your motivational inspiration that time. Thank you very much. And finally, I had a session to interact with our foreign faculty. I thought that this would be great that our participants, if they can interact with the faculty member and the foreign faculty, especially Professor Michael Chikandas we have, and Dias is also here. If anyone want to interact, though you can unmute and ask the question, but for your clarity, I would like to name someone who has joined us from remote. So for some of the senior faculty, we have Professor K.P. Kocher, we have from All India Institute of Medical Science, Delhi. Professor Chikandas is a very renowned institution, and Madam is HOD of Department of Physiology. She is working, and we are glad that she was a participant of this course. I am very happy. I came to know that she joining our course since last first lecture to the last, and appreciate it. I would invite Professor K.P. Kocher, if you are able to hear me, please unmute and put your words. Yes, Madam, can you hear me? Okay, so meanwhile, I will invite the Professor Muniz Garg, if he is there, is former Dean, Faculty of Pharmaceutical Science, and he works on natural product derived from the plants. If he is there, he can unmute and interact with the faculty.
Yeah, Dr. Tiwari, can you hear me?
Yes, you are audible, sir, please.
Yeah, thank you, Dr. Tiwari, for giving the opportunity, and good evening to you all. Rather, good morning to Professor Chikindas. And
Thank you.
it was pleasure listening to you now for 2 minutes, and it was very interesting. I am working in medicinal plants on various aspects and for their clinical efficacy and standardization and quality control. Now, you might have covered a few things related to this biota and their significance in medicinal plants and agriculture. And but my question is a bit different, and I would like to ask if you can help us. In many times, microbiota or microbes, they are creating problems on the medicinal plants during the storage and while cultivating. They are having the growth on the plants, so they are reducing their efficacy and safety profile. So can you explain a little bit that how we can prevent those nasty bacteria, or how to get rid of these things so that we can prevent the unwanted reactions on the medicinal plants? Professor.
Well, thank you for your...
Dr. Chikindas, I would suggest you interact in the last, so you probably have all the queries and you can address in a very short time.
Oh, okay. Well, we can do a Q&A now, and
Okay, if you want, you can.
Yeah, sure. I think it will be more convenient also for those who are asking questions. Right. Briefly, my understanding from what you are saying is how to control microorganisms in the plant-derived products, not necessarily in the growing plants, because there are several phases. Growing, and I guess it's a controlled growth, hydroponic or greenhouse environment. Then you have harvesting, then you have processing. And the processing, since you mentioned the word plant, it's not a plant extract, it's probably, I don't know, dried or, certain parts of the plant. So the control of non-desired microorganisms, it starts with the growth conditions. So, right, this is the first phase where you want to have microorganisms. And you want to have the microorganisms that actually assist with removal of the pathogens and the microorganisms that otherwise not desirable, which can cause spoilage eventually. This is where we need to talk about plant probiotics. And I mentioned in my animal section that unfortunately due to the time limitation, we cannot talk about plant probiotics, but this is something that is being developed now. They're mostly coming from bacilli, some lactobacilli, and some from Pseudomonas. But later stages, it's a non-invasive, mild processing. And this is where you cannot use one method. There should be combinations of methods, all right, which may work and should work synergistically. Like, for instance, I don't know, mild heat which will not jeopardize the qualities of your plant product in a combination with, the very same bacteriaceae. They work. I can envision it as a small 6 months up to 1 year project, something given which can be shaped into the master degree research project, in collaboration with Dr. Tiwari, the targeted microorganisms isolated, and then to administer, different types of physical, chemical, and biological factors, mostly focused on physical and biological to remove chemistry from this scenario. The first thing I envision, the mild heat, steam, for instance, it's just, thinking cloud, if you wish. Steam, it can be the wet steam, the dry steam, in a combination with the bacteriocin spraying, and it can be delivered through the same nozzle, through the same delivery system, and looking at the elimination or control of the non-desired organisms. Short answer, yes, it can be done. I think it's a very interesting, good project, and I suggest, to communicate more with Dr. Tiwari and me, and we can actually design this project, and hopefully you guys can submit it for, kind of a small grant, or for the not necessarily for the full size research, but also for educational grant, to have a student benefiting as well. Thank you.
Thank you, Dr. Chikindas. We have next faculty, Dr. Ritu Yadav from Department of Genetics. She want to interact. Dr. Ritu, can you hear me?
Hello, Dr. Tiwari?
Yes, you are audible.
Yes. It is my great pleasure to be the part of this valedictory session. Namaskar, sir. Myself, Dr. Ritu, Associate Professor in the Department of Genetics. I am working in the field of epigenetics, particularly in the cervical cancer, to find out the differential expression of different genes in the cancer. Sir, I just want to know about we can say how the probiotics, and we can say the purity of the probiotics can help in the area of cancer. Am I audible, sir?
Yes. Thank you very much for your question. It's a very interesting area. And of course, we cannot generalize. It's all called cancer, but you certainly know that cancers come in a variety of origins, and they go in a different ways. It's like proliferation, but what causes this proliferation of the cells? So the causative agents are different, and so how to address the causative agents, it depends on the agent. What I am more familiar with, and the reason for that is that at some point in time I was thinking of putting together a proposal, and actually it was shaped, but it never flied. The health-promoting bacteria in colorectal cancer. Why colorectal cancer? Because it is one of the broadly present cancers in the world. Number one, so it requires significant attention. Number two, because colorectal cancer, when it is properly and early diagnosed, it can be limited, it can be controlled. And number three is that we know from the literature published that there is a correlation in the changes of the gut microbiota and the transition to colorectal cancer. So there is a good indication based on the preliminary foundation that microorganisms can actually work as modulators. In a particular example of the cancer, I'm trying to be very careful not to spread to other types of cancer. It all depends on what your specific objective. What kind of cancer? And you mentioned epicervical cancer. So I'm almost, okay, 99.9% certain that with the epicervical scenarios, we have a dramatic change in microbiota, right? We know what is happening. So there are two things: predisposition and the treatment. So in the case of predisposition, I think it is incredibly important to look at the factors that are considered as factors, in the group of predisposition factors, and to look at the prophylactic options. This is where I am 100% sure that proper selection of probiotics may help, and this is where most definitely an opportunity for the academic grants. But I would also encourage to establish connections with Lalimand, which is #1 company working in the area of women's health. All right. Definitely either small sponsorship or collaboration or something like that. Treatment. I mentioned briefly today that some bacteriocins were reported as active specifically against cancer cells. I've never done these kind of studies myself. I did studies on the herpes simplex virus. I did studies on HIV. Niacin and subcalosin are working at very high concentrations and the specific targeting in the mass. I think there is some really good possibility there, but I have, anybody will ask me where I will put my money to. Bacteriocins, possibly. Peptidamimetics, most likely. So I would probably try to see, and in India there is a lot of very good research going on in the area of peptidamimetics. So this is where I would try to see, when it comes to the cure, when it comes to the treatment. But I think, on the scale of the country as India, with growing population, women's health is one of the most important things, among others. And if prophylactic treatment may emerge, based on inexpensive health-promoting bacteria, it's a win-win situation. So 2 objectives, 2 strategies, and I think both of them are equally important and very interesting. Thank you.
Yeah. Thank you so much.
Thank you, Dr. Chikindas. Hopefully it was beneficial for our faculty. For any query in detail, you can write an email and he will reply.
Right, and if you're interested, we can try to approach, the company I mentioned and to see, if they're interested, or, with Dr. Tibari, who is always my collaborator, we can think about any possible future developments there as well.
Just one, Dr. Kiral, yes. Sir,
Yes.
we all know cervical cancer is just started from the initial to final stages. There is a very long time period.
Yes.
Do you think there is a microenvironment have the basic played a role in the progression of the cancer, or we can plan on the suspected females and plus cervical cancer female?
Yes,
Are you
yes.
fine for that?
Yes, most definitely, yes. Most definitely, yes, and that is why I'm saying that it is very important and very interesting. This can be modeled. The time when I started working with it, and I was using also the MUDTEX epivaginal model. You may either used it or heard of this model, which is a fantastic, ex vivo model to study these scenarios mimicking vaginal epithelia. What I found when I was also working with the epicervical isolated cells and isolated tissues, actually pathogens, they hate healthy cells. They're afraid of healthy cells. They're trying to move away from healthy cells. So if we have the environment healthy, it can stand for itself even without bacteria. But when the environment is disrupted, this is where, healthy microorganisms can and should help. Nowadays, we have, the time when I was working, to make the single microorganism biofilm was a huge challenge. Nowadays, we know examples of multimicrobial biofilms. We can grow biofilms on the surface of the epicervical cells as well. It is possible if they are properly treated. So, or on the synthetic surface, it can be modeled even better, more mechanistic studies. And then you are absolutely right when mentioning to look at the profile of the microorganisms in women at different stages, right? And then to see what kind of microorganisms are they and how they can be controlled, how these microbiota can be modulated. And obviously to look at comparison with the healthy subjects, which particular species of lactobacilli are there and here, and then to put an emphasis on these kind of microorganisms and to see how they can control the potentially pathogenic bacteria in the environment. I think you just pointed to a very interesting project.
Thank you so much.
Thank you.
Thank you, Dr. Tiwari.
Thank you very much.
Interaction with the sir. Thank you, sir. Thank you so much.
Thank you.
Next is Dr. Neelam from Department of Genetics.
Good evening, everyone present over here. Am I audible?
Yes, you are.
Thank you, Dr. Tiwari, for giving me the opportunity to interact with Dr. M.L. Chikandar. Sir, myself, Dr. Neelam Sehrawat, I am Associate Professor in Department of Genetics and I am working in the malaria. I am basically working in the transmission blocking vaccine development based upon the vector. There are two ways for the vaccine development. Either we can develop the vaccine on the plasmodium based or we can develop the vaccine by vector. So we are working on the mosquito system. So we are taking the candidate antigen gene of the mosquito and by multi-epitope approach by using immunoinformatics, we are constructing the vaccine for the transmission blocking of the malaria parasite. As well as I am also working on the plant protein for the logistical activity against the mosquitoes, particularly Anopheles that is causing the malaria, and also against the QLEX for the dengue and Aedes. So this is about my area of research. My question to you, sir, for example, in case of drugs used for the plasmodium control or for the treatment of the malaria, for example, chloroquine, I have read that with the chloroquine, there is, if we give the probiotic along with the drug, there is, you can say there is significant reduction in the parasitemia. So I want to ask, is there any other antibiotic also available, particularly name of that antibiotic that can be given along with the malarial drug?
Okay, to answer short, I know what kind of papers you refer to. I haven't seen other examples. At least I didn't come across in the literature. And my guess would be, since you are asking, you didn't find them either. So the short answer, we don't know because they were not reported. All right, that's number one. Number two, can there be others? I think there can be. And I think the important thing, what we see here, and again, is at the level of the gut feeling, if you wish. I think it has to do with the immunomodulation. Yeah. It's definitely immunomodulation. So if you can find, or, since you're in the genetics, and one of the sessions was dedicated, to the engineering of probiotics. And if you were there, I'm personally in favor of laboratory evolution. I think it's, I don't like it. I think laboratory evolution is better in the sense that it gives you more opportunities to explore what is inside by selecting what is on outside. So you're selecting by biomarkers, you don't care how they pop up. You just need them, all right? And they can come up in a variety of ways, okay? So what I would do, I would probably coin a project aiming at improvement of immunomodulatory functions of the selected probiotic organisms, all right, which will be acting at a certain site, targeted site where you see them acting, all right? So this is a very good standalone research project, number one. Number two, it's the first phase. The second phase is to make sure that this probiotic is tolerant to the drugs that are used, starting with the already known, and then moving to, you can also select your own drug of choice and, through the laboratory evolution to keep the biomarkers, but also to identify the forms that will tolerate this kind of drug in the environment. That's what I would do. So it's a combination, if you wish, of the natural classical selection with a twist of novel method and classical microbiology. But my heart tells me that this is about immunomodulation. From what I remember from the literature, I did not come across any other drugs than what was already mentioned. I don't know if you and I missed something or they're not reported yet, but basically that's an approach that I would use. And of course, the interesting thing will be also with the delivery, how to cleverly design the delivery, but you can talk about it with your engineers.
Okay, sir. Thank you very much.
Thank
you,
you.
Dr. Tiwari, for giving me the opportunity to interact with sir. Thank you, Dr. Tiwari. Good night.
You are most welcome. Actually, Professor Chikandas, I have received few names. They are eager to interact with us. The reason they are bothering you, and probably we are penning you because you have already given delivered lecture. We have Dr. K.K. Sharma with us. He is head department of microbiology here, and also serving as associate dean in the R&D section actually in our university. I invite Dr. K.K. Sharma. He also worked with the gut microbiota and is a very good researcher we have with us in our university. We want to interact with you. Dr. Sharma, over to you.
Thank you, Dr. Santosh. Am I audible?
Yes, Dr. K.K. you are.
Yeah. Okay. Good morning, Michael Chikandas. Good morning.
Hello.
And hello. Yeah. So it's a pleasure to see you. And I have been co-PI or collaborator in a joint project during COVID with you. And I was very excited to interact personally regarding few queries which we have regarding gut microbiota. I basically work in the area of microbiome and biome of the ulcerative colitis patient, and how this microbiota shifts when a person migrates from rural area to urban area, like lifestyle disorder. And further, my area is metabolic enzyme present in the gut microbiota and how it affects negatively or positively in UC, ulcerative colitis. My query is, or I want to know your viewpoint regarding the doses of probiotic. How to define the doses? Because right now there is a big problem of that. Another thing is, how does it take care of dysbiosis? For example, if doses are not appropriate, it can lead to dysbiosis and it can change the keystone species present in our gut. This is one. Another one is your view about the future of personalized probiotic. As you know, everybody has a personalized gut microbiota. So, what is the future of personalized probiotic? Is it possible in near future?
All right, thank you. I have three questions, somewhat interconnected: the doses, the combating of dysbiosis, and personalized probiotics. So with regard to the doses, you may remember over the course of our sessions, we were talking about several examples where some authors show the dose-dependent action. I think it's almost like a rule of thumb that if we are able to deliver something like 10 to the 5th, 10 to the 6th cells, we do see the effect, the positive effect, providing that the probiotic works positively. Everything below 10 to the 5th, most likely you will see no effect. If you go above 10 to the 6th, 10 to the 7th, 10 to the 9th, 10 to the 9th, most definitely there will be diarrhea and some adverse negative effect. All right.
Okay.
So the safe zone is in the area between 10 to the 5th and 10 to the 6th. This is where is a safe zone. All right. With dysbiosis, this is the mysterious field, absolutely mysterious field. And you may remember when we were talking about probiotics for pediatrics, I was mentioning an absolutely fantastic work by GIAN when they designed the probiotic formulation for the newborn child who passed through several surgeries and had just Candida albicans in the gut, not a single other microorganism, purely Candida albicans. And they managed with a formulation containing lactobacilli and bifida to wash away Candida from the gut environment and to replace with normal microorganisms, which became a booster for the future development of multi-species normal microbiome. So, how is that happening? I doubt anybody can answer you the question. I think the most important part here is if you observe that dysbiosis, to identify which microorganisms, and you can sample the environment, which microorganisms dominate instead of the normal microbiota being dominating, and then to identify the probiotic bacteria which can actually work as potent colonizers in the strain replacement or species replacement scenario. This can be done. I think it's not just can be done, it is being done. And this is what is used often with the fecal transplantation. And of course, we have to keep in mind that fecal transplants are not considered probiotics per se. All right, but this can be done. Mechanisms, many, but nothing is reported as being specific. Question number 3, personalized. I think for humans, definitely we will see more positive results, more positive outcomes in personalized, and I think it will be coming. I think that personalized medicine will emerge sooner or later, and I can judge by the number of NIH projects that are sponsored over the years where, once in a while, personalized medical approach is appearing as the proposals, and some of them are granted with financial support. So personalized approach, most definitely, yes. And especially if you remember, there were some cases, clinical trials with lactobacillus GG, no positive outcome whatsoever. Why? We don't know why, because when people are getting negative results, most of the cases they don't study why they received negative results, what went wrong. Was it the scenario? Was it the administration? Was it something wrong with the diet or with the human subjects? We don't know. So I think that personalized, right now it is most expensive, but I do know several companies internationally in many countries, what they do, they collect, individuals are approaching these companies, they collect the fecal samples, they identify the profile of microorganisms, which is indirect and not very accurate, but still some kind of level of approximation of an answer of what is actually going in the gut of this person. The non-invasive method. Of course, you can do colonoscopy, you can collect the sample from the side, but that's a different story altogether. And then, based on these analyses and the individual studies, these companies suggest these specifically designed formulations of probiotic bacteria for a specific user. These companies I know in Europe, in the US, and they're growing in numbers and they're relatively small. So, even university may have a spin-off as a startup company, doing these kind of things. And I'm saying it loud because I know that, senior university managers are with us today. So I think this will be very rewarding, and this may create a very good foundation for the future. And of course, for those who will approach this kind of individual personalized treatment in the near future. But most definitely the answer is yes. Thank you.
Thank you. Thank you very much.
Thank you, Dr. Chikandas, and thank you,
Dr.
KK. Yeah. Thank you, KK, for interacting with our honorable foreign faculty. Thank you very much.
No, we are getting late, so there are a lot of queries, but it's not possible in single meeting. So
I will forward his email ID and you can direct email.
There are a lot of queries because we know microbiota is so complex. Can't just have one discussion. It's very complex.
We need another session to start with.
We need that.
Thank you, Dr. Chikandas, for very elaborately answering the queries of our faculty member. Some of the participants, they want to give feedback in a very quick because we are getting already late. Dr. Malashri, Pratiksha, and Harspriya, if you are connected, just they want to give the feedback of the course. Dr. Malashri, are you connected? Pratiksha from Maharashtra, Harspriya from Karnataka. I don't think she is. So Hina, are you there?
Yes, sir. Am I audible?
Yes, you are.
Good evening, everyone. This has been an absolutely amazing and enlightening workshop because of the mixture of short explanations with a lot of practical exercises and virtual lab. Industrial visits makes it more enthusiastic for all the research scholars, where it fulfills the need of both learning and experiencing the techniques. I'm also grateful for the structural and informative sessions of Dr. Chikandas, which helps us to connect with the lab experimentation to the application. Huge gratitude to Dr. Chikandas and Dr. Santosa for all that you have shared with us about how to imply in the lab work to the industrial scale-ups. Overall, thank you very much, sir, for the smoothness with which the workshop was conducted. I honestly believe that I can't put into words the importance of workshop to me and to all the research scholars. Thank you so much.
Thank you, Hina. Thank you very much. Subham would like to say quickly, please. Subham from Delhi. Subham, can you hear me?
Connection.
Yes, Subham. It is audible.
Hello.
Please continue.
Good evening, everyone. This is Shubham Sharma, PhD scholar, South Campus, University of Delhi. Today I would like to share my experience for the coursework. Firstly, I would like to express my great gratitude for the opportunity to be a part of this learning experience. This course content was not only instructive but also presented in a way that made complex concepts more understandable. I would like to thank Dr. Chikandas as he demonstrated a deep knowledge of subject matter and their passion for teaching us was evident. I would like to thank GIAN Course. I have observed remarkable engagement in discussions, in insightful contributions to the assignments, and a genuine eagerness to grasp the course contents. In conclusion, I believe this course has been a valuable investment in my education as my research topic is much related to the probiotics as it is the probiotic consortium development for the oral cancer, and I would look forward to applying the knowledge gained, and I'm comfortable with that it would contribute to the success in my future. Thank you so much, sir.
Thank you, Subham. Next is any Manoj and Indu. If you are there, please unmute and very quickly.
Hello, I am audible, sir.
You are audible, please go ahead.
Good morning, sir. First of all, thank you for giving a valuable and important lecture during the GIAN program, and thank you, sir, for organizing such type of program. I, Dr. Manoj Kumar Yadav from Department of Genetics. Sir, I am very thankful to you for giving a broad area of probiotic and different topics like oral health and genital health and other like bacteriocin purification, mode of action, and their application in food. Therefore, your lecture are very much knowledgeable and give a direction for the future to introduce ourselves in next level of research in this field. Thank you, sir.
Thank you.
Am I audible, sir?
Indu, yes, you are audible.
Yes, sir. Good morning, sir. First of all, I would like to thank you, sir, for the really significant effort you have put in this course. All sessions were very informative. I think all the sessions were very definitely enlightening all the participants as well. So my name is Indu Kumari under the supervision of Dr. Santosh Kumar Tiwari, sir. A big thank you for this opportunity, sir. So basically my work is on genetic characterization of Lactobacillus plantarum and Enterococcus cereus for production of bacteriocin, in which I have done the molecular identification of both the probiotic strains, their plasmid profiling and antibiotic resistance. After that, I checked the strains of the bacteriocin production, whether it's associated to plasmid or main chromosomes, in which I did plasmid curing using plasmid curing agents. Also, further mutant analysis was done using the mutagens. I have the mutagens in which I have done the over 1,000 colonies of each treatment was screened for bacteriocin activity. So basically, I want to ask one question regarding the as we all know that a number of strategies were used to analyze the gene involved in audited bacteriocin productions. So, sir, basically, I want to ask that how can I further characterize the bacteriocin gene? Is only by comparing the mutant and wild type on the basis of the bacteriocin amplicon obtained after PCR is enough or not, or I need to do something else?
Well, thank you for your question. Thank you for your good words. I appreciate it. I think you're on the right path with doing what you are doing now, and working with Dr. Tiwari, I wouldn't dream of anything better. So you're in a great team for sure. One thing important is, and you're doing the right thing, no question asked. One thing important, however, is to make sure that you are not missing something. So yeah. I would suggest, if it is possible, and of course, funds are always coming as a limiting factor. Can you sequence, your strain and to see what kind of potential genes, most definitely, potential transcripts, most definitely. And then linking structure to function, you're just doing the right thing. There is nothing can be added. But and you know, another thing is, and you're working with lactobacilli, so I wouldn't expect much of the problems there with the expression. But when I was working with the mutacin 1 and mutacin 2 from Streptococcus mutus, and that was the effort with the University of Alabama in Birmingham many years ago. My very good collaborator was Jan Novak at that time, and there were genes, but at that time the genetic analysis platform was not as advanced as it is now. We knew that there must be bacteriocin, but we couldn't find it, and Jan Novak spent a year to design a medium that would allow for bacteriocin production in the liquid medium. The bacteriocin was beautifully produced on the solid surface, and we do know that there are many regulatory factors that influence bacteriocin production. You know, the type of cultivation is one of them, pH, oxygen, temperature, many. So the important thing not to miss something, because we know that some of the strains can produce multiple bacteriocins. That's number one. Number two, to answer your question, if you can do anything else, I think your strategy is perfectly all right. But, you know, genetic analysis, comparative analysis to see if there are potential operating frames that may be coding for some bacteriocins, that's obviously the must. Thank you. And good luck.
Yeah, thank you, sir. So just 1 second also. Sir, is there means that if one bacteria producing several bacteriocins, like like enterocin gene, enterocin bacteriocin, and also some other bacteriocin?
It is possible. We know examples, like for instance, streptococci, multibacteriocin strains of streptococci that are employed now by the followers of Dr. John Tagg, even, you know, to the point of developing commercial products. Yeah, it is possible.
Okay. Thank you so much, sir. Thank you very much.
Indu, we can get this answer on the email also.
Yeah.
The last one was the Pallavi. Would you like to say something very quickly? Pallavi, you are not audible. All right, so
I'm Pallavi.
there is some. No, it's not audible.
Of Dr. Santosh Kumar Tiwari. Sir, very firstly, I
think there is a problem with the connection.
To express my gratitude
Pallavi,
for
it's okay.
It's all the informative data you have.
I think.
Sir, now am I audible?
No, you are muted. Unmute and say. I think it's fine. It's fine. They are saying the similar thing. It's open to the audience if you want to say something. Otherwise, I am going to end this session. If someone wants to say, can quickly unmute and say. If there was a beautiful interaction, great with the faculty member and most of the research scholars also they highlighted. And at the onset, the time has come to say bye to Dr. Chikindas with a very wet eye and painful heart. I am not able to say how to say thanks, because the kind of contribution he has done and he is doing with me is enormously unbelievable. So I'm very thankful to Professor Michael Chikindas for his precious time taken with us and answer most of the questions, organize this course in a very systematic manner. The motivation was from our Honorable Vice Chancellor, Professor Rajbir Singh Ji. He provided all the infrastructure facility with us, right from the internet connectivity, the resources. There were at least 50 human resources. They were working for this way, that way, this should be right, this should be right. So we are organizing this way, and all the supports from the Vice Chancellor administration came. I'm very thankful to all the personality from there, and also the chief guest of the day, Professor J.P. Yadav, he came, and he was actually instrumental. He was working with us as a senior faculty member in the department, but on deputation he had joined as a vice chancellor in the nearby university. I am very thankful to him, sir. And Dean Academic Affairs, Professor Surendra Kumar, Dean College Development Council, they had on board here on the dais. They all motivated us for organizing this program. Dean Faculty of Life Science, Professor Rajesh Dhankar, was also with us to understand, analyze the things and do the things along with the HOD Department of Genetics. Dean R&D, Professor Arun Nanda, and Associate Dean K.K. Sharma was also involved in this. Local coordinator of GIAN, Professor Narsimhan, who was actually implementing the administrative part of our course to do in a very organized manner. I am very thankful to all the head of department, directors of our faculty, and really not last but not the least, all the participants. Without their respected audience, it was not possible to have our honorable foreign faculty, Professor Michael Chikandas, and technical supports from University Computer Center, and also the funding agency, GIAN National Coordinator, Ministry of Education, Government of India, for providing us that opportunity to get the grant and implement this course in the reality. Also, we had some local media coverage also. I send you to in Hindi and English somewhere. They cover us to the national level and put it on the newspaper. I'm very thankful to them also. So at the end, Professor Michael Chikandas, we apologize for your extra time and actually painful. I can understand your sitting since last 3 hours. It's really painful, but whenever I meet, we will have a cup of tea and tea from India to make all pain relievable. Thank you very much.
Thank you very much for your nice words. It was a very, very interesting and fruitful experience for me. Again, many thanks to the senior management and the granting agency, the support staff, the participants. And I want to say that, over the course of our sessions and today as well, several things were communicated which I identify as possible projects, standalone research projects that can be funded. Please don't leave it alone. Work on these things. Communicate with Dr. Tiwari. We can communicate together. Of course, even if you are from different departments, different schools across the country, we're here to help each other. We're here to produce some knowledge that will be beneficial for the students, beneficial for the education, and beneficial for the society at large. So this is our duty, this is our responsibility, and this is our desire. And this is why we're here. Dr. Tiwari, if you will have media coverage, please pass it to me. I will pass it to our international department. They will certainly like it. And with these, we're closing our sessions, but I'm not saying farewell because you and I are still working actively together, and we will continue collaborating as we did in the past. So thank you very much, everyone, and hopefully we'll see you sometime in the future. Thank you.
Thank you, and bye. Namaste from India.
Bye.